Welcome, everyone. It's me again. If you were here for the previous session, I'm Michelle Gray, chair of the SAB Scientific Advisory Board for HDSA. Like I said previously, I'm a researcher. I have a research lab at the University of Alabama, Birmingham, where I study Huntington's disease pathogenesis in mice using a mouse model. So today, it is indeed my pleasure to have Dr. Cha come here today to talk to us in this section, drug discovery from idea to treatment. I think it's a very important session and especially important for this particular group, Huntington's disease patients and their families. You know, as a scientist, I go to various events across my campus and I talk to fundraisers and the development people, and they say, do we have a cure yet? You know, like, what are you doing in the lab? Shouldn't we have a cure by now? And so, you know, when is this stuff being translated into real therapies that we can put into people? And I often have to say this is a very long and difficult process. And so we have Dr. Cha here today, who I think probably better than a lot of us understand this very well. Dr. Cha is presently the Vice President and Global Head of Translational Medicine for Neuroscience at Novartis. But in his former life, he was an academic clinician at Massachusetts General Hospital, where he saw movement disorders patients, including Huntington's disease patients. He, I think, is no stranger to most of us in the HDSA community because he is past chair of the HDSA board, as well as the HDSA Scientific Advisory Board. He's still serving on our Scientific Advisory Board and as well as past on the Executive Committee for HDSA. So I think that Dr. Cha is going to give us some very good information today, teach us a little bit about how we go from scientific ideas and hypotheses that we're testing in laboratories to really trying to translate those into the clinic and develop therapies for diseases like Huntington's disease. Thank you for coming.
Got to get my water. Thanks, Michelle. All right. Guys, you made it to the home stretch. This is the last session before Gala, and if you're wanting to just relax and just have an easy session, you've come to the wrong session because we're going to make you work for it. First, the disclosure, I work for Novartis, but I'm not talking about Novartis stuff today. But I don't want this to be about me, right? So let's make it about us, right? So I'm going to ask you to think along with me. How are we going to solve this problem? So there's going to be required participation, including from those of you who are live streaming it. There'll be written assignments. But, all right, but what's the best way to teach somebody something? What's that? Have them teach it, okay. Hands-on, all right, that's what we're going to do, not what you signed up for. But let's start up, okay, what do we want from research? Okay, let's let the ideas flow. A cure, okay, what else? What else might we want from research? Understanding, okay. Information? What do you want research to do for you? Solve the problem. What problem, though? Let's get specific. What? Slow down the symptoms? Movements? What? Treatments, okay. What kind of treatments? For HD, okay. What part of HD? Cognitive, okay. Psychiatric, all right. So there's a lot of things that, you know, I'm stalling for time, but basically, you know, basically we want a cure, and that means different things to different people, right? There are lots of aspects to this, and we could have kept on going, but, you know, maybe delay the onset or control symptoms like irritability or thinking is a big problem. The movements can be really problematic, right? So maybe a treatment to maintain weight or depression, right? So there are lots of things. Swallowing, that's something, boy, if we had better stuff for that. Falls, slow the progression, that's a big one. Memory, right? So this may be on our wish list. So okay, so that's our motivation. How are we going to do it? All right, so how do we get from an idea, and you've heard a lot of ideas, especially in the research symposium, okay? Lots of ideas. How are we going to turn that into a treatment, okay? And this is going to be all of you. We're going to do this together. So what do we want to get to? Well, our finish line, treatments for HD, right? And they might be treatments attacking those symptoms that we just went through. But what needs to happen for a treatment to be available for HD patients? Trials. Okay. Well, actually, so this is our finish line, right? Get effective treatments to HD patients. What do we need to do to get to this point? Human research, right? Okay. Actually, to get a treatment approved, we have to have an approval from an agency, like the FDA in the U.S., right? And what do we got to do to get that approval? Anyone? Bueller? Okay, we need trials. Yep. Okay, so you need pivotal trials that will convince the FDA or the EMA, if you're in Europe, that your drug is safe and effective, right? And how are you going to do that? You're going to do a clinical trial, all right? So the reason you need the trials is without the trials, you don't get the approval. Without the approval, you don't get the drug, all right? And the trials that get your approval are your pivotal efficacy trial. Those are phase 3 trials. I'll explain what those mean. But to get to phase 2, you got to get to phase 3, you got to get to phase 1 and phase 2. So to get to phase 3, you got to get to phase 2. To get to phase 2, you got to get to phase 1. I didn't change those to white. Okay, so we will go through these definitions, but you guys are already drug development experts. You understand the phases, right? All of these are clinical trials. What does clinical mean? What does people? Okay? Clinical trials is not rats. Clinical trials are in people. Okay? So these are clinical trials, and we group them into phase 1, phase 2, phase 3. We'll talk about what that means. All right? But this is what we need to do. All right. Anyone? How are we going to do this? Okay, let's start off with a little thought exercise, because you've seen this, right? You've seen this in the newspaper or on your Google News feed. Here's the headline: a new breakthrough for HD. Oh, I'm going to read this, right? Okay, so this sounds interesting. Okay, I'll read this. Professor Big Brain from Big Deal University has published a study in the Journal of Super Important Science identifying a potential treatment for Huntington's disease. All right. HD mice in which the bad guy gene was switched off had delayed onset of abnormal symptoms and had prolonged survival. Okay, you've seen this story, right? Exactly. All right, so do we have a treatment? Oh, all right, so what does that tell us? What do we think now? You read that, what do we do next? What's that? It's all about the guy. Okay, so there's maybe a new target, right? Maybe a new target, maybe a new strategy. What else? You read this in the paper. What do we do next? Let's say you're a company. Let's say you're Novartis. What do we do with this? Okay, I mean this is some laboratory geek. I am one. It's done something with mice, and I've done stuff to mice, and it looks like something worked. What's that? From mice to human? Okay, so all right, 15-yard penalty on myself for saying guess what I'm thinking, but what does this tell us? What does this tell us? What does this really tell us? We have a little news blurb. It's 2 paragraphs long, and there's no detail, but you see this right? So it says, look, this might be a target for HD, this bad guy protein that I never heard of before, but I just read it in Google News Alerts. But that experiment, they switched it off in mice. Well, that doesn't seem like that's going to work in people, right? That might be really difficult. Is there another way we can do this? You got a bad guy protein? Bueller? Anyone? This is a bad protein. If you knock it out, the mice don't have HD. Yeah, well, so you'd want to know that. There's a lot of question, you know, with every research, every good piece of research raises more questions than it answers, right? But maybe you can come up with a therapy if there's a way to block that protein, right? If you come up with a medication that blocks the protein, that might work, right? So could we design a drug to block bad guys? So what's the first thing? Okay, this has landed in our shop. We're a drug company, guys, all right? So this has come across our shop. What do we do? What's the first thing we're going to do? It's called step 1. What do you want? Advertise? You don't advertise till you have something. What's that? Say that again? Talk to the researcher. I will accept that. Actually, the first thing you got to do is replicate the result. Okay, dirty little secret. A lot of the stuff that appears in journals, much of it cannot be reproduced. Okay, we're a small drug company. You know, actually, this is about the size of a very small drug company. We can't afford to waste our resources on stuff that's not real. First thing we're going to do is replicate that study. See if we can do it in our own labs. We got smart people in the room. If they can't do it, then it might not be real. Okay? We're going to replicate the result. And hey, we do it, and maybe it does work. So guess what? That gets us to step number 2. Excellent. Good. Step 2. So now what do you want to do? So we've replicated the result, and actually Professor Big Name result looks like it could be real. What do you want to do now? What's that? Safety study. Safety study. Okay, good. Tox? Yeah, tox is short for toxicology, which means what? Well, okay, so what's that? Well, we remember what the original report is. They genetically turned off this protein. What? Okay, so we could figure out what the protein does, right? Yeah, we have a bad actor protein. It's doing bad stuff. Okay. Yeah, I mean, I would want to. This is an assassination, strategy. I would just take the bad guy out, right? And this, we've heard lots of versions of the assassination strategy today. By the Huntington lowering, we're going to take out Huntington, right? So if we had a chemical that could block this protein, that might be a good thing. Now, how are you going to do that? Okay, so we're going to chemistry lab, right? What you do is you start testing chemicals in Petri dishes, right? And you see if that chemical can block that protein, right? So there's a lot of chemistry involved. There's a lot of assays. There are a lot of long words that I know that we're not going to talk about. But basically, you do these little experiments in Petri dishes, and what's happening now is we don't use Petri dishes anymore. We've miniaturized them into these well plates. So on the bottom left, that's a 6-well plate. Then you have a 12-well plate to 24. The industry standard is now 1536. Okay, what does that mean? Yeah, so instead of testing one chemical at a time, you can test 1,500 chemicals at a time per plate. Okay, when you start screening compounds, I'm just going to show you what it kind of looks like, right? So you run these little experiments, and they're essentially like little mini Petri dishes all right together. You might have a result that looks like this, all right? So in these different wells, you have different results. Question?
So what if it turns out it's only a cocktail of those that's going to work?
Yeah, so you won't do that you won't see that in your first screen. So it's a good.
Can you get back to that? Can you get later in the process?
You can absolutely get back to that, but what you're going to do is it's a great question. What if only cocktail? But how many compounds are you going to screen? And the answer is like gazillions. Not gazillions, but probably millions. So what happens is, and you're going to screen them in these 1536 well plates, so many at a time, and people don't do it, robots do it. Robots do it, right? So why are you screening 1 million compounds? Because most of them are not going to work. Most of them are going to be toxic, or they're not going to block the activity. So you start with what are called chemical libraries. A library is just a set of different chemicals, and it's usually about 500,000 to 1 million different compounds that you start off with, and you screen them robotically, and you start to see which ones of those are having the activity that you want. So we have a protein that we want to block. We set up an assay that was those yellow colored wells that we showed you, and then we want to see which of these 1 million compounds is going to have this activity. All right, so you do your initial screen, and it really is about 1 million. It's got 10,000 here, but that's really, and then a subset of those will have like some activity, and you'll advance them further, and there's a funnel. But you can think of this whole process as really kind of like salmon swimming upstream. You know, you have a lot, right? And at every stage you're going to lose some, and you know there's going to be some attrition. But what you'd like to do is avoid some of the bad ones. Okay, so we got a target. We're screening compounds now. Okay, so we're screening for the activity. What's the activity? Well, we've got a protein that we think is the bad guy. We're going to see, is there a drug that can take this bad guy out? We've got a little assay, a little test that'll tell us if it's working. And we look for the strongest compound. So, and again, robots do this, right? And I'm happy to submit to the will of my robot overlords. But the things that work, just to introduce some terminology, are called hits. If you get something that works, that's a hit, right? So, and you might have heard this in some of the talks today. We're talking about hit to lead chemistry. This is what happens: is you screen your 1 million compounds. Most of them won't do anything, but some of them will. Okay, and I keep saying compounds. Compounds. What is a compound? It's just a type of chemical. Okay, each compound is a different chemical, and some of these chemicals are pretty complicated. Now we got a bunch of hits. Are we done? No, yeah, you knew that. What do we got to do now? Yeah, well, you won't know this. You got to optimize. You do know this. Holy crap. You went to the wow. So it turns out the hits that come out of your screen are never a drug. They're never a drug. You screen 1 million compounds, you got 16 hits, you're happy. None of those will be a drug because they won't be safe for humans, but it's a good starting point. So you take your hit and you make several versions. This is medicinal chemistry, and there are loads of chemists who are working in these companies, and they take that hit and they'll make 20 versions of that hit, or 100 versions of that hit, and then okay, now we got like different versions of the hits. Are we done? Oh yeah, so it's like we've identified hits and made several lead series. Each lead series is like several different versions. It's like pepperoni pizza, mushroom pizza, pepper and onion pizza, various flavors of that hit. Are we done? Exactly. Exactly right. Right. So some of those versions will maybe lose the activity, right? Yeah, sorry, repeating, that's what you're saying. The question is like you're making different versions of the hit and changing different parts of that. And yeah, that's exactly right. But when you change different parts of the molecule, that might change the activity that got it into that screening round, right? So we just made, 20 different versions of each hit. What do you want to do now? Rescreen, right? So then you rescreen and say, okay, did we improve the activity of some of these, or did we lose the activity? And then it'll tell you which parts of the molecule you can mess with, right? By the way, this is how Me Too drugs are made, is you take something that you know that works and you change it a little bit, you make it a little bit better, and it's, it's how you go from Coke to Pepsi. This is how pharma does it. But okay, so now we got a bunch of compounds, and want to introduce some definitions here, just the way we use it. Preclinical means animals, and test tubes. Clinical means people, okay? So you hear about we're at preclinical stage, we're ready for clinical trials. Clinical just means people, preclinical are critters, okay? So we now have a bunch of stuff to test, all right? We've got a bunch of chemicals, any of which could be a drug, and I'll tell you, most of them are not going to be a drug. So what do you want to do now? And we might have 100 of these compounds now. What do you want to do? Yeah, so we're going to start messing around with animals and seeing what this does to animals. And it's preclinical because it's animals, right? Efficacy and safety. Safety first. Does it kill the animals? Because if it kills the animals, we don't get to put it into people, okay? Right? And does it work in animals? Okay, so if you have an animal model of your disease, you want to see that it's efficacious. But you start with safety always. Always, always, always. And all drugs are poisons. All drugs are poisons, right? It's just a matter of dose. So you have to understand for each of these drugs how much you can give before you start seeing problems. You will have toxicity, and it could be toxicity on your heart or your kidneys or your liver for all these drugs. You just don't know what level is. So yeah, we have to study extensively toxicity. This takes a lot of time. It takes a lot of time because some of the toxicity doesn't come out until several weeks of treatment, right? So we have to, before we get to play in humans, we have to know that it's safe in animals. Understanding that no drug is going to be absolutely safe, but understanding how much you can give before you start seeing problems in animals, right? Takes a long time. Okay, so we're taking animals and largely rats, but not just rats. And in fact, you got to test it in 2 different species. You can't just do it in rats, right? Are we ready for the clinic? Okay, what do we got to do to test this now in people? Can we just do it? No, we have to get permission. We have to get permission from a regulatory agency. And it's the FDA in the US, it's the EMA, which is the European FDA in Europe. And you have to send in a really long application, which takes a lot of work, showing that you have a reason to believe that this compound has a reason to be effective and that it's safe, because you've done all this, about a year plus of toxicity studies in animals, and that you know a lot about it. And you know a dose of 1 gives no problems, but a dose of 10 gives these problems, and a dose of 100 kills the animals, or something to that effect. So you have to provide a huge stack of data about what you know about your drug. And then you also have to say, here's what we plan to do in people. And the FDA will say, yes, we agree, or no, you're not allowed to do this. Same with the EMA. So you have to make an application, and this is a big hurdle, right, once you get into people. And the bar is high, and it should be high, right? We're putting new molecules, new chemicals into people that have never been in people. Question in the back.
What is happening with the FDA now? How long is the time frame? And have they fired FDA people? So it's less staff now?
The question is what's happening with the FDA and how long is the time frame and have they fired FDA people? I'll take the last question first. Yes, they fired FDA people. The turnaround time, though, is pretty standard, and you can get an answer within 30 days. Within 30 days. Yeah. FDA, remarkably, is very efficient. They're very efficient, and if they tell you they're going to give you an answer, they give you an answer. And the day before, we sit around our telephones waiting for that phone call. They're extremely efficient, which is surprising, yeah. But they will tell you what they don't like, and they'll give you green light, and same for the EMA also. So it's actually a remarkably transparent process. You can go to www.fda.gov. The whole thing is spelled out. You can look it up, and it's remarkably kind of easy to understand. But look, the point is you can't take any chemical that your chemist just made and put it into people, right? And I think that's right. We have to have a real good idea that it's going to be safe. And let's acknowledge that we're jumping from critters into people, right? And that can be kind of informative, but there are some toxicities that only occur in people, so you might not see anything. It's risky, right? Question in the back.
I have a question about the process through the FDA.
Yep.
You say they turn around and give you an answer promptly, but at the beginning of this application, they say, yes, you have to perform all these tests and these kind of things for the next 5 years, then we'll consider it?
Yep. And it's all on their website. It's all on their website. And it's very detailed what's required for this initial application. This is not an approval application. This is just a green light to start the studies. Phase 1. Right? Yes, question.
How much does potential efficacy have to do with whether or not the green light study?
Yeah.
Is it so much that
The question is, how much is potential efficacy to green light the study? It's important, right? Because if there's no unmet medical need, the FDA say we're not doing it. There are some human conditions that you cannot. There's not an adequate animal model to demonstrate that. So, for some things, it's really impossible to demonstrate that it will work in an animal model. So you have to provide a rationale of why you think your drug, your potential drug, would work, and that's based on. It's a long way of answering. The short answer: it matters. It has to. If it's successful, it has to make a difference. So, but really, the question. It's a good one. The primary criteria at this point is safety. Do you know that it's safe, right? All right. We're in people. We got the green light. We're putting it into people. Actually, we got the green light from the EMA, or so we're doing it in Belgium. Belgium's even quicker. We're doing it in Belgium. What's the first thing that you want to know? Is it safe? Absolutely. Safety, safety, safety. Is it safe? Are people dropping dead? Or are they tolerating it? Do they have nausea? You want to know what it's doing to your people, right? And we start with a really low dose, and then we go up to a slightly higher dose, etc. So now we're into the clinical trial phases. You've seen this, right? They're all on the FDA website. Phase 1. We're in phase 1, right? So right there. This is, phase 1 is typically in healthy volunteers, and it's all about safety and tolerability. So it's not just, does it damage them, how do people tolerate it? And these are small studies. As has been mentioned, if you have a new drug, you don't want to expose many people to a drug that you don't really understand the safety. So you start with small numbers, and based on what happens, and these people are really very closely observed. Typically, these volunteers, and a lot of them are college students, are brought into a clinical research site, and they're kept on premise. They're not allowed to leave, right? And you're collecting everything. You're collecting blood, other bodily fluids, because you want to know how it's affecting how it's affecting them, and also how you want to make sure, you want to understand if you give a pill, how much of it gets into the bloodstream and the like. So you really study intensively these patients in phase 1. So people get paid. The question is, do people volunteer? Yeah, it is voluntary. People do get paid. Interesting thing. So people get compensated for their time. There are strict limits on how much you can pay your healthy volunteers. There are strict limits. Now think about that. Now you heard earlier about enroll and how a lot of clinical trials fail because they don't enroll the clinical trial. Now think about that. There's a huge number of clinical trials that never get completed because you don't enroll enough people. So this is a question that never gets answered, right? So on the one hand, for a pharmaceutical company, it would be great if we could just pay people and get them into the trial. The reason that that's not allowed is it's seen as coercive. If you dangle a lot of money in front of people, then they're not able to make the judgments about something that's potentially affecting their health. So there are really limits on how much you can compensate people. You're allowed to basically compensate them for their time, but people do not get rich off these studies and I think that's a good limitation, right? Because you don't want to just exploit people who might really need the money. So let's unpack we're in phase 1, right, guys? By the way, now that we made it into phase 1, we've had a champagne party. We're happy, all right? We're doing good. All right. What happens in phase 1? We've already said this: safety and tolerability in healthy volunteers, small doses, then gradually escalated in several cohorts. So you have a group of 8 patients or 8 volunteers. You give them a little bit of this drug, and you watch them, and then you review all their lab data, their blood tests and everything, and their EKGs, and then you'll review all that data, and if that all looks okay, then you might give a dose to the next group of 8 at a slightly higher dose, and that's called dose escalation. So this is like adding salt to soup. You don't start off with what you think is going to be the final amount. You start off with a little bit, increase it gradually, checking every time. This is intensive study, so they're in units and they're having bloods drawn, and they get compensated for that. Typically, because if there are other, the question is, do they have to be on other medications? And typically, no other medications. Sorry. Yeah. So if you're trying to understand your medication, sometimes if a subject, and we call them subjects if they don't have the disease, a healthy volunteer is a subject, and a patient is someone with a disease. So the subjects, if they're taking another medication, it can complicate your understanding of your own drug. And there are drug-drug interactions too. So typically these people are required to not be on any medications. Yes? Yeah, so we'll talk about blinding a little bit. The question is about blinding. In phase 1, you typically are blinded, okay? And that means there are people in phase 1 who are getting placebo. Okay, if you're doing safety and efficacy, or safety and tolerability, why do you need people to get placebo? What's that? Yeah, so if the side effect is itching or nausea, and everyone reports it, and you have no one on placebo, then you don't know if it's the drug or it's just everyone. So the placebo group, the people who are not receiving the active drug, helps you interpret the side effects. So the other type of phase 1 studies are phase 1b studies. They are phase 1 studies done in patients, not subjects. So done not in healthy volunteers, but in patients with the disease. The phase 1b studies are typically done in cancer. So in oncology studies, they skip the healthy volunteer part. They go right into cancer patients because some of the oncology drugs are super nasty. So much so that they don't want to expose healthy volunteers to that stuff, right? But they will expose cancer patients who might get some benefit also. So that's phase 1b. How are we doing? Oh, great. Phase 1 studies show that your drug is safe and well tolerated in healthy volunteers. That's a huge benchmark, guys. Good job. What do you want to do next? Phase 2 is the correct answer. What do we want to know, though? Yeah, we now know that it's safe, and now we can get to the question, does it work, right? That's phase 2. There we go. What happens in phase 2? It's like, does it work? All right. So to get to phase 2, you have to have gotten through phase 1. See, this stuff is not so difficult, right? We made it through phase 1. We're now at phase 2. But what happens in phase 2 is, does it work? All right. And so you have some idea of how the drug behaves, what the pharmacokinetics, that's PK, and is it hitting the target? Is it hitting the bad guy target? Typically, this is the first time the drug is hitting the patients, those patients for that disease. So phase 1, even if it's for Huntington's disease, phase 1 might be in people who don't have Huntington's in their families, right? But phase 2 is where you're getting into patients with Huntington's. And we're looking for efficacy. What does efficacy mean? Does it work? Yeah. So let's just do some definitions. Phase 2A is typically, you've got 2 groups. You've got the drug versus a placebo. A placebo is kind of an inert substance, right? Phase 2B is where you do dose ranging. You'll test multiple different doses, and that's kind of what dose is required for it to work. So 2A, 2B. So how do you know if your drug is effective? Okay, one answer is you have a benchmark or something you can measure. That's good. Examine the patient. Okay, good. What's that? I'm sorry. Go over your data. Okay. So what data are we going to collect? All these are correct, but what data are we going to collect? You didn't know there's going to be a quiz today, right? All right. So yeah, okay, right. So the comment from the back, did it make the protein go away? And we had a comment over here, it depends on what your efficacy marker is. Because, for example, maybe it does make the bad guy protein go away, but maybe it doesn't change your Huntington symptoms at all. So, so how do you decide if something works really matters, and, you could imagine a phase 2 study where actually the objective is to measure bad guy, and if it went away, then that's success. But another phase 2 study might be, okay, can they walk better, or do they have fewer movements? That's a different, so what your benchmark is matters. Question.
How do you deal with the placebo effect?
The question is, how do you deal with the placebo effect? You tell me. That's how this is going to work.
People believe that they are better and they are on placebo.
Yeah, so the comment, I'm just repeating it so people can hear, that people believe when they're getting a placebo that they're better. And I'll tell you, it's true. If you take people into a clinical study and they're in the placebo arm, so they're not getting any real drug, they always do better, a lot better, sometimes shockingly better. What's that? For a little while, right. So that could complicate things, because we could give somebody our drug and they get a lot better, but it might be placebo, right? It might just be the effect that they're in the study. How do we know it's our drug that's really doing it? Time? Yeah, okay, so look, what we're working our way towards is like, what do we need to see? We're going to spend on this phase 2A trial between $5 to $10 million, all right? So let's consider carefully what we're doing, right? We're probably going to get to something like this, a randomized, placebo-controlled, double-blind phase 2A trial. So I just wanted to jargon it up a little bit, but we'll just walk through some of these terms. Randomized, what does that mean? That means the patients coming into the trial have a equal chance of being assigned to the placebo group or the active group. Why does that matter? Well, it turns out that if you non-randomly assort people, and let's say you put all your worst patients with the most severe symptoms into the placebo group, and you put your least affected patients into your treatment group, it's going to look like your treatment group did a lot better, right? Because you didn't randomly assort them. The way to get equal groups is you randomly assign them. Placebo is important. It's an inactive substance that resembles the experimental drug. It looks just like the pill for your drug. So the patient cannot distinguish the drug from the placebo, but it doesn't have anything active in it, right? So this is what's referred to as a sugar pill, although most of the time it's not really sugar. Double blind, what does that mean? That means the patient doesn't know, that's a single blind, and the investigator, the doctor, does not know which assignment. Okay, double blind is important, that neither the research subject or the investigator knows which assignment. Why do we need to do this? Bias. Somebody tell me about bias. Yeah. Yeah, so look, if we had so bias can work both ways, but let's imagine that your doctor, who you've had a tremendous relationship for 10 years, says, I've got this great drug, I think it's really going to help, let's see if it works. You're going to want it to work. that doctor, he or she wants it to work for you, right? Guess what? You will do better. You will do better. So the problem with that is that's not the real effect. We want to know, does it really work, right? And phase 2A, we went over this. This is primary efficacy. So let's get back to how are we going to decide if we won or not, right? So we are in Chicago, right? So about last year, right? What's your primary endpoint? This is Game 7 of the 2016 World Series, right? There's a lot of numbers up there. It was extra innings, if you remember, right? But the primary endpoint in this game, it's been decided, is the score at the end, all right? That's how you're going to decide. Oh, this isn't Game 7. Oh, but this is, which game? Game 6. Okay, but this is the primary endpoint for Game 6. Okay, we're going to look at the score at the end, okay? So if you look at the primary endpoint, you could conclude Cubbies win, fly the W, right? Everyone's okay with that, right? Let me talk to you about post-hoc analysis. Post-hoc is Latin for after the thing. So you can look at the same game, close game, and say, yeah, I know we got our asses kicked if you're Cleveland, okay? But if you look at these innings, okay, we won these innings. We won these innings, okay? So you can always do a post-hoc analysis and fish out a result that you're going to like, but that's not the same as missing on your primary endpoint, okay? So it matters what your primary endpoint is. I mean, it could be knocking down bad guy protein. It could be improvement in motor function, but you just have to stick to your primary endpoint. When you're reading these press releases, be very skeptical of studies that miss their primary endpoint, because they can always go back and say, yeah, but we won the 6th inning, right? All right. Conclusion, yeah. So, yeah, we won the 4th through the 8th inning. It's like, yeah, that's not. All right. Anyway, so we're marching our way through these trials. Any idea of how much they cost? Phase 1 studies? Phase 2? Yeah, phase 1 around $3 to $4 million. Phase 2 around $5 to $10 million. Phase 3, we're talking $20 to $100 million. So it adds up. It's expensive.
How does it break down by salary and by
I think I actually got rid of that slide. But the majority of his clinical trial costs, right, is clinical trial costs. So the question was, how does that break down in terms of where the money goes? Most of that is for the study visit, okay? So the study visit, let's say a company like mine is running a trial. We will run a trial in, let's say, 82 different sites in 15 countries, okay? And we're hoping that each of those countries can enroll something like 12 patients. And in that trial, if it's a long trial, they might be coming in monthly for 6 months. Every time they come into the clinic and they do those assessments. People here have been in clinical trials, right? Okay, right. So every time he came in and they did assessments and they administered things, that site got paid. That site got paid. So that's the majority of the cost right there. Okay, so we've done our phase 2 now, okay? After phase 2, we have some evidence of efficacy. What do you want to do next? What phase do you want to go to next? I know you know this. Phase 3. Okay, so what happens in phase 3, right? So these are the trials that you need to show to the FDA, right? It's the phase 3 trials that get you your approval. But you don't get to phase 3 unless you are successful in phase 2. Okay, so these are what's called the pivotal trials that are used for registration. And they are typically much larger. Why are they larger? Right, because the FDA, the EMA, they want to be sure. If you have a trial that works in 40 patients, that's good, but it could be a fluke, right? So your phase 3 trials might be much larger, might be 200 patients. Okay, and the more patients, the more cost. So that's a big driver of why these things are so expensive. So you take some information that you learned from phase 2. Number one, is there some hint of efficacy? Also, you've done your phase 2B trial, which shows you which dose works the best. You take your best dose from phase 2B, you bring it into phase 3. Does that make sense? You guys are doing great. Okay, so here's the thing that the FDA will not accept. They don't care if you knock down bad guy protein. That will not approve your drug. So in the MS world, MS now has 16 approved drugs. 16 approved drugs in MS. And the way that drug companies develop drugs in MS is they use the MRI because they know if the MRI scan will show them if their drug is working. You cannot use the MRI results to get your drug registered. The FDA says the patient doesn't care what their MRI looks like. The patient cares if they can get up and down the stairs, if they can tie their shoelaces. So it has to be functional, okay? It can't be a biomarker to get your drug approved. The biomarker can tell you, are we on the right track, right? Okay, so, you know, that's a little bit of a roadmap, but if we do this right, these are some of the steps, we can reach the goal. Let me pause there. I know I've dumped a lot of stuff on you. We'll open it up for questions. In the back. The question is, how long do phase 1, 2, and 3 trials typically last? It depends a lot on the disease that you're studying, and the jargon is the indication. So phase 1 is typically about a year. Phase 2 is typically, let's say, 2 years, and then phase 3 can be 3 to 5 years. You know, why is phase 3 so much longer? Because you have recruitment issues, and it's the number of patients and how quickly you can get them into the trial, which is the major determinant of how quickly you can do these trials. Question in the back.
You said some of these drugs that your company runs in 15 different countries.
Yep.
What countries do you find to be the fastest and for using for drug companies?
Yeah, so the question is, you know, when you're running a trial in multiple different countries, which are the fastest? And it's a combination of different things, because in all of these are regulatory agencies, okay? So the regulatory agency turnaround time is really critical. EMA is very fast, FDA is very fast. The Chinese agency, the CFDA, very slow. So that's a big disincentive to going there, although they're trying to change that. So there's the regulatory agencies, which are governmental, but then it's the individual sites, you know, and they're largely, you know, university clinics. We know which ones have done well in the past, and those are the ones we like to go to. The ones, as a general answer, most of the trials get done in the U.S. and Western Europe. Eastern Europe recently, in the past 10 years, has really increased their number of clinical trials. So there are a lot of clinical trials going into the former Soviet Union with some improvement. I mean, it varies, but right now I'd say the Western European countries and U.S. are kind of the fastest. Question, this gentleman.
I know the signal study, the FDA came out and said they were going to fast track that.
Yep.
What does that actually mean? Is it getting better?
A fast track means like an expedited review, but it doesn't actually lower the bar in terms of the amount of proof they have to. But it makes things go a little bit faster. So it's helpful to get the fast track, right? Ricardo? Yeah. Yeah, so the question is, with diseases that have slow progression, does that change? It absolutely does. So if you do a trial for blood pressure, you've got an agent which changes blood pressure. You can do that trial where every patient is on treatment for a month, and you'll get a real answer. if you're doing treatment for Alzheimer's disease, you can't see a change in a month, right? And Huntington's, I think, is the same way. Huntington's does not get worse day to day, doesn't get worse week to week, maybe month to month, maybe year to year. So this is kind of the difference between a symptomatic treatment and a disease-modifying treatment. And I like to think about cars and rusting cars, right? So a symptomatic treatment and a rusting car might be a spray paint job. You can do that very quickly, and it can look better, but it doesn't stop the underlying process. The neurodegenerative process is like in Alzheimer's or in Huntington's is like rust in that it moves slowly. And if you have a rusting car and you sprayed it with rust inhibitor, you would not be able to tell the next day if you had made a difference. You might not tell the next week. So we're running a trial now in Alzheimer's where some patients will be treated for 7 years. Yeah, the question is what's the ideal duration for a study in HD? So I'm going to make everyone here answer that. How long should let's say a disease modification study in HD, how long should that be? What's that? 18 months? Okay, where's that coming from? 18 months. Why 18 months? Okay, let me just repeat that so people can hear, right? Because HD goes up and down, you don't want to have too short of an observation time because you might get an inaccurate result. So what would be helpful is to understand, on average, how fast does HD progress? Where do you get this type of information? Yeah, enroll HD, track HD, predict, right? So these observational studies are super, super important, right? Once you understand in a large number of people, right? And let's acknowledge that individuals are really different, right? So we're not going to base a whole trial on the one HD patient that you know because they're going to be different from their sibling even, right? So we got to look at a bunch of patients, understand, okay, how much change can we expect in patients with repeat numbers in the 40s over a year or over 6 months or over 18 months? So let's not go through the actual calculation, but that's the kind of information. So natural history observation is hugely important, and it'll tell you how to power your study. Let's talk about power. It's not an ego thing, right? But how do you power it? This means you need to have enough patients in your trial so that when you get the result, you know it's real. So you don't have just one patient because that patient could have good days or bad days. You want to have enough number of patients so that you believe the result, and it has to be long enough so that you also believe the result. And you get that power calculations from the natural history studies. Does that make sense? Once you start to understand how this disease goes, then you can say, okay, if we had something that slowed it by 50%, how long would, let's talk about a fleet of rusting cars, okay? And we have a rust inhibitor that slows rust by 50%. How long would you have to treat those cars and observe them to see the benefit? Yeah. Question in the back.
So Jonathan asks, are you excited about the NFL biomarkers becoming a real game changer in clinical trials?
The question is about NFL, which everyone remembers is neurofilament light. I'm hugely excited. Just to remind people if they missed it, there's a report that came out a couple of weeks ago from the Track HD study that neurofilament light, and neurofilament light is a protein which is in brain cells. When these brain cells get damaged, this protein gets released into the bloodstream, okay? So neurofilament light is, in a way, a measure of brain damage. What you see is that it increases over the course of HD. So it is another way to measure disease progression. So very excited about that. And if we have something that really stops, that slows down the disease progression, we ought to see that reflected in an improvement NFL, neurofilament light. The other reason we're excited about neurofilament light chain is it turns out it's not only a special marker in HD, that's just the latest news. It's also been implicated in stroke, MS, Alzheimer's, a disease called progressive supranuclear palsy. So this gets to be a little bit exciting where we can have one test that could work for multiple diseases. We do not have that in neurology yet. There's not any test that we can use for multiple diseases. Neurofilament light chain is now looking like one of those candidates. But the real excitement is it gives us something to measure, right? And we could imagine doing a trial. It's like, okay, if it's disease modification, maybe the first thing that'll happen is it'll improve the neurofilament light chain. Other questions? Yes.
What's the incentive for drug companies to work on drugs for a rare disease, given the expense of all the research?
Yeah, it's a good question. So the question is, what's the incentive for companies to work on rare disease? So I'm going to answer a question with a question. What do you think the cost of a new drug is? What's the total cost? How much? A billion. We have a billion. A billion's close. It's 1.3 billion by some estimates. By other estimates, more like $4 billion. $4 billion. I used to think this was. What's that? No, that's just the development. So the reason it's so high is you're rolling up the cost of all the drugs that fail. Okay? So each individual successful drug does not cost $1 billion, but you don't know which ones are going to win. But the total average cost, if you look at all the R&D costs divided by the number of drugs that actually made it to the market, is actually on the order of $4 billion. Hugely expensive. Obviously points to some systemic problems that we could do better. What's the incentive for companies to get into rare diseases is, number one, is it unmet medical need? I can tell you that most companies are really driven by unmet medical need. Is there science that we can understand? Okay. I think if you listen carefully, the companies that we've heard about are interested in HD, but they're also thinking, what can we learn from HD for other diseases, right? If we can crack this disease, then with our assassination strategy, can we get to other genetic diseases? But most companies are really doing it because there's a need. There are some companies that are trying to get into ultra-rare markets and charge the hell out of people. I don't think that's a sustainable strategy, frankly. I think it's unpopular. Yeah. Guys, you did so good today. You guys did really, really good. All right. Thanks.